So I'm very pleased to be able to introduce Anjali to give tonight's lecture. So she is a research leader in paleobiology and the Dean of Postgraduate Education at the Natural History Museum, and also an honorary professor of paleobiology at University College London. So obviously these appointments don't happen by chance. She has an incredibly impressive research record. And I don't want to give spoilers as to what the talk is going to be about, but she's published more than 130 papers on the evolution of different groups of animals, from insects to dinosaurs, but is especially focused on the evolution of mammals. And I'm particularly envious that she seems to have fieldwork that involves going from Svalbard to Madagascar, and is currently leading expeditions in Argentina and India. And so with nothing else, it's my pleasure.
Thank you very much. Well, thank you very much. It's a real pleasure to give this lecture and to really kick off this new part of our regular program and to really highlight the new fellows, because of course the fellowship is the core of the society. So as Stuart mentioned, I do quite a lot of field work, and up here you can on the video you can see some of the highlights of the field work that we do in my lab. But I'm going to start off first by talking just a little bit about some of the highlights out of a different sort, which are the highlights of the Linnean Society. And to really point out, as I mentioned, this society is, a fellowship at its core. And as a fellows only event, I can really point out some of the amazing opportunities that are available to all of you, and also to ask you to participate in some of the things that we have coming forward. So first of all, we have an incredible variety of awards and medals, from early career researchers and students through to people who contributed to the understanding of science or the appreciation of natural history, to art. And so please do take a look at the medals and awards site, where you'll be able to see how to nominate some of your colleagues. You must be a fellow to nominate people, so this is why this is an ideal audience to have this conversation with. The deadline is coming up very quickly, so just at the end of next week on September 30th. So please do take a look at that and nominate some of your amazing students and colleagues. Now, the other thing I want to highlight is our journals. I think I certainly, before I became a member of council, didn't quite appreciate how important the journals are to the society, not only for being a primary way of us communicating the amazing work that the fellowship, as well as the broader scientific community, do, but also as a key source of income for the society, allowing us to do all the great works that we do, including supporting this building and all of our events and education programs. Now, this year, some of you, hopefully all of you, know that we launched a new journal, the Evolutionary Journal, as well as maintaining our 3 excellent journals, the biological, zoological, and botanical journals. So I really encourage all of you to send your best work to these journals. I think I can count at least 10 papers that I've sent in over the years, or actually that have been published over the years. I'm sure I've sent in more that weren't published. And so please do consider submitting your work. Now, some of you will also know, of course, that the first description of evolution by natural selection, this joint publication by Darwin and Wallace, were published in the Linnean Society Journal. So, I think that's quite a good history to publish in. If you find that intimidating, however, I just want to point out something I noticed recently. Now, we give lots of talks and tours to government officials and other of the high and mighty, and we often pull out this copy of that first publication that came out the year before The Origin of Species. And I noticed in the last time this little footnote at the bottom, and maybe some of you have already noticed this, but I hadn't, which was, I love this because this is literally the first publication of the theory of evolution. And it comes with this caveat, not written with care, the basis of the most important discovery in the history of biology, in my opinion. But it's also, I think, is a reminder to take the pressure off of ourselves a little bit and be a bit confident about our work. But I will say that it is probably an accurate reflection of the talk you're about to hear. This is certainly not a talk I would submit for publications. Parts of it are very experimental, parts are a bit personal, but hopefully you will enjoy it. And so to start off with, normally I would start a talk by introducing my research program and some of the key questions that I find to be of interest. But I'm going to start at a different point. In this case, I'm going to start off with, how did I get here? Because one of the things I love about the Linnean Society is that it is an incredibly broad tent. People from all different backgrounds, all different interests, all different areas of expertise come to the Linnean Society. And in that way, it's really distinct from any other group that I'm part of. And so when I think about, how did I end up here? And how did I even end up being a paleontologist, which is a question I get asked many, many, many times. And it's one that I think most people are usually surprised by my answer, because I did not start off like one of these kids. This was never me, I don't even know that I ever thought about dinosaurs as a child. And it absolutely was not this. I actually only saw Jurassic Park for the first time during the pandemic. So this wasn't how I got sucked into paleontology. I had a very different origin story that I think is reflected in the work that I do today. So this is where I come from. This is Detroit, Michigan. Some of you will recognize it. It's most known for being the home of the big three automakers. Some of you will recognize one of those, Ford isn't usually a company that you think of as being a radical pro-environmental company. But of course, this week in this country, Ford is obviously rebuking our government, along with many other people rebuking our government, for their really abhorrent U-turn on their environmental commitments. But that's not really what Detroit is famous for. But Detroit is where I was born and raised. But actually, Detroit itself is probably not the most important place that I experienced as a child. These are my parents. Not in Detroit, not a lot of elephants. There are some elephants, but not many in Detroit. But when I was around 4 years old, my parents, who were in the US on visas, their visas expired, and their renewal was rejected. Something that probably some people can sympathize with. And so when I was 4, for the first time for me, but obviously not the first time for my parents, we went to India. And so I spent a year of my life at this point in a place called Madhya Pradesh. This is the central Indian provinces. And this was actually a life-changing moment for me, a life-changing period for me, because the part of India that my family is from is the part of India that this book is based in. So the central part of India is where something like half of the world's tigers live. Certainly back in the 70s, it was incredibly covered with forests. It was really beautiful. You'd see wildlife everywhere. Sadly, that's not really the case anymore, but there is still quite a lot of forest cover in the area. And so when I was 4 years old, it was the first time I saw a scene like this. This was actually a scene, a photograph that we took the last time I was in India when I took my kids to the same place. But it was really these moments that I remember as the formative experiences of my life and led me down this path of being absolutely passionate about the natural world. And that really has continued. I think a lot of paleontologists, a lot of evolutionary biologists, a lot of natural historians really pride themselves on being obsessed with really unusual or unique creatures. But for me, I'll be honest, it's always been tigers. And as I learned more about deep time and actually got to experience evolutionary biology and paleontology, which is something that I didn't actually know was a real job until I was well into university, to be honest. I realized that everything that I loved about the natural world and from early childhood was actually just even more interesting when you added in evolution. And this goes, of course, for cats as well. And so this has been, I think, the continuing passion of my life. And I just want to pause for a moment and talk about that word, passion, because I think it is something that we at the Linnean Society all share. It's this incredible passion for the natural world. Again, regardless of what angle we look at it from, or what our experience or expertise or knowledge base for the natural world is, we all share this deep commitment and passion for the natural world. And sometimes that is viewed as being at odds with being a scientist like myself, right? So we can see this even in Darwin's own descriptions of a dispassionate judgment. And of course, dispassionate here is referring to being objective, making good decisions, being not being influenced by emotions. But I think it's often really misunderstood when people think about science or people who study the natural world in all of its forms as being people who are not passionate about that world. And yet I think all of us know that it's the exact opposite. In fact, we are really driven by our emotions and our feelings for this wonderful planet and life on it. And of course, Darwin, even though he would say things like this, scientific man ought to have no wishes, no affections, a heart of stone. He also wrote things like this. What does this say here? I'm dying by inches for not having anybody to talk to about insects. This is not a man who lacks passion. Maybe, lacks stability, but definitely not just completely lacking passion. And so when I give my scientific talks, this is what I say my research program is about. What intrinsic and extrinsic factors influence the tempo and mode of evolution of organismal form? But of course, the passionate side of me says, why do animals look like this? That is really what I'm interested in. That's what I've always been interested in. From the first time I looked at tigers in the wild to today. Why does the world look the way it does? Why does life on Earth look the way that it does? And this is obviously an incredibly interesting topic that nobody cannot be excited about. I think I might be wrong, but I think I'm right. And when I think about diversity and the diversity of life, I really think about it on really large scales, right? I like looking at these really big patterns and trying to understand how do we get these patterns? Where do these patterns actually come from? And when we look across the world today, we can of course see some of the really obvious patterns, right? When we look across the planet, we know that there's far more species in the tropics and in the poles. When we look across time, so we could look through deep time, we know that there's periods in the past where you have really high standing diversity that's maintained for relatively long periods, punctuated by really rapid changes in species numbers. So we can have huge extinctions that decimate global flora and fauna. We have huge radiations that really quickly produce new species and new forms. And when we look across the tree of life, we know that there are some clades that are incredibly diverse, whereas other clades are relatively depauperate. And really what I want to know is why. Now, when I think about these questions, I think about them probably slightly differently than a lot of people, because all those examples that I just gave you, they're very familiar, and so they're a good way to think about these questions, but they're all based inherently on species numbers, which is a straightforward thing to compare, numbers of species. But actually, I'm not thinking about species numbers. I'm thinking about variety of species forms, variety, essentially. And this is an area that I refer to as biodiversity phenomics, really trying to capture the phenomic diversity of the global biota. So this term phenomics might be unfamiliar to some people. It's a slightly fancy way of saying, morphology or anatomy. We're trying to rival the genomicist here. But really, it's more than that. Because an organism's phenome is really the sum total of their observable physical characteristics. So not just their morphology, it includes their behavior, their physiology, their development, all these aspects that we can observe in species unified under the title of phenome. And these are the characteristics that are really important to understand because these are the very things that mediate their interactions with their environment, with other individuals in their own species, and with other species. And as a paleontologist, for me, it's actually the only way I can even try to estimate a lot of those qualities about ecology and life history, because we'll never be able to observe those things in life. Now, thinking about this, how we can capture diversity and why we get diversity in the amazing range that we see today and in the past is a really interesting question, but it's also a hard one to access or to just know where to begin with. And so sometimes it's useful to actually flip things around and start with the opposite, start with things that aren't so diverse, which I like because it brings me back to my favorite of all animals, cats. And cats are incredibly interesting because they are really not very diverse, if we're honest. There are many species around today, and they actually all kind of look the same. They have different colored coats, but I usually look at skeletons, and when it comes to their skeletons, there's not a huge amount of difference. Now, not everybody agrees with me. As I learned, one of the areas, in addition to the passion for natural world, another area where you can often find lots of passion is on Twitter. And I, without really thinking very much about it, I think a few years ago now, or a year or two ago, posted just a tweet about cat diversity or their lack of diversity in their phenotype. And I was just thinking about this in terms of what it means in terms of adaptive peaks and things like that, and said, well, maybe cats just nailed it. Maybe they have it exactly right. They have just the right morphology, and they only need to get bigger or smaller, and they don't need to change anything else about themselves. I turned off my phone, went out for the day with my family, came back and found out that people have opinions on the internet. Lots of them, it turns out. And so there are tons and tons of people who were, what about sharks? What about bats? What about this? What about that? Overwhelmingly missing my point that all those groups are great. They're very diverse. They have lots of different forms. And my point was that cats don't. And that is interesting. Right. And if I was feeling feistier, I might say maybe they have so many different forms because they're not good at anything. But I wasn't feeling feisty. I was trying to be dispassionate, which you can see in some of my responses here. But the truth is, cats are really weird. They are the only tetrapods, the only limbed vertebrates that get above about 50 kilos and can still go into a crouch. All other groups of vertebrates, when they get above that size, go straight-legged, what we call graviportal. They go very straight-legged. If you think of your average artiodactyl or a hoofed mammal or something like that. Cats are the only ones that can get so big, hundreds of kilos, and still crouch down. And in fact, their posture does not meaningfully change across this huge body size range. And that is a really interesting thing. In fact, we had an entire grant just trying to figure out how they do it. I don't know that we actually figured it out, but we at least quantified some patterns. But this really comes down to development. Well, at least how this pattern is maintained comes down to development. And we can see this really clearly when we look at cat versus dog development in something like the skull, for example. So this is a figure from a paper we published many years ago now. But if you look at the skull of cats across ontogeny, so through their development, from a kitten to an adult, they basically stay about the same. We call it isometry, just change with body size, but no change in proportions. Whereas if you look at dogs, they actually show incredible change through ontogeny in the proportions of their skull. And this is, of course, why, if you really want to mess with cats in terms of breeding, make them look as silly as possible, this is the best you can do. You can take their hair off. Other than that, a cat is going to look like a cat. Dogs, on the other hand, there's all kinds of ways to make them into ridiculous animals that should never survive in the wild. And this really comes down to these developmental differences. And of course, this is just reiterating what Conrad Waddington pointed out really nicely many years ago now, that the evolution of organisms must really be regarded as the evolution of developmental systems. And so even though I'm a paleontologist, my work has really always been rooted in this idea of trying to understand how we can access information about development and how it influences evolution when all we have is morphology. And sometimes we might have some development. But we can see that even in really closely related species, like these sister species here, a really, really minor change in developmental timing, in this case, just delaying sexual or having sexual maturity before earlier in their ontogeny, creates completely different adult morphotypes in otherwise closely related species. And if we didn't know anything about development, we might look at this and think, this is a huge, huge shift in morphology. But actually, it is just a trivial shift in development that creates these massive changes. So how can we use this? And how can we try and access this? This is something that's really driven my research. And I'll talk a little bit later with some examples of where we see that playing out. Now, there's another aspect of the cat and dog example, and I promise I won't really talk about cats tonight, but I do like them, even though I only have a dog actually now, which is not just that kind of timing of the way the skull changes or the body changes through development, but also actually looking at how traits change together. And this has actually been probably the primary area of my research over the past 20 some years, which is looking at the relationships among different parts of the morphology. And so this is actually, again, something we can take all the way back to Darwin. So Darwin was actually really obsessed with white cats with different colored eyes or with blue eyes. That's because they were almost always deaf, maybe always deaf, actually. And so he knew that there must be some linkage between these traits. And of course there is, but didn't have the capacity to really understand the full pathway then. But we know now that when we see integrated phenotypes or integrated aspects of phenotype, it reflects associations during development or in the genotype among these different traits. And actually, these relationships are incredibly important for understanding how morphology or how anatomy actually evolves, and therefore for understanding how diversity evolves. So when we look at these, if we look at all the possible forms that can exist in the world, we know that very, very few of them actually do. So this figure on the right here, it's kind of what we call a Raupian morphospace. It's showing the variety of shell shapes that we see among mollusks, and how the way that they develop, the way that they coil, really limits the possibilities for what forms can actually evolve. And that really comes down to the fact that these traits are covarying. As they develop, they're limiting the possible morphospace that they can occupy. Now, this other figure on the side, the one that looks like Saturn, is just a really simplistic simulation that I've done, where the red dots are just, if we had started off with a simple shape, a skull in this case, and just let it evolve over many, many generations, the red dots represent all the possible morphospace, so the possible different forms that we would imagine would evolve in the absence of any trait relationships, any linkages, developmental or genetic or functional even, among individual traits. The black dots, on the other hand, are a different simulation where all we do is impose a very realistic amount of interactions among the different traits, such that when one changes, it makes another one change as well. And you can see that that immediately limits the way that species evolve. It limits the variety of species that we see. And it forces a lot of different forms to evolve over and over again because there are limitations in how they evolve. So what's interesting is that this developmental patterning actually makes things like convergences, which we tend to find really interesting. We talk about them a lot. We treat them as though they're something quite unusual. Actually, because of development, they should be the expectation. And so we coined this model a few years ago called the fly in the tube model, which is about the evolutionary expectations of integrated phenotypes. Again, integration being those covariations or correlations among different traits that arise from genetic and developmental relationships and patterning. And basically the tube is what development forces all of evolution into. Now within that tube, actually, we see morphology and evolution zipping around, but it stays within a relatively constrained space that's limited by the initial kind of developmental patterning that arose, what we call canalization. And again, this gives rise to convergences. So here we have on one side a thylacine, a marsupial wolf or Tasmanian tiger. On the other side, we have just your bog standard wolf, placental wolf. Probably some of you can tell them apart, but they are not easy to tell apart. But these are two species that are separated by, say 160, maybe 180 million years of evolution, 160 probably, million years of evolution. And yet they look incredibly similar because at the core of it, mammals share a very common developmental patterning. And so given that, not every single kind of form that we can imagine can arise, actually forms are relatively limited in certain areas of morphospace. And so this is the kind of thing that's really been at the core of my research over the past few decades, is trying to take all these different elements and really try to understand why life looks the way that it does, both today and in the past. And this is, of course, where paleontological data becomes really interesting, because we know that the vast majority of life on Earth that has ever lived is extinct, right? What's around today is a tiny, tiny, tiny proportion of life that's ever existed. And we know that the life that is around today isn't representative of the life that has existed in the past. So if we look at, what the larger clade that birds are part of looked like, 100 million years ago, we're going to be looking at things like these dinosaurs, right? And we can also see that, things that are just entirely extinct clades, like these marine reptiles or these pterosaurs, not to mention some of the really unusual forms that we see at the base of certain clades, are completely different from what we would imagine if we just looked at modern diversity and tried to walk it back through our understanding of how things are related to each other. And you don't even have to go back millions of years to really move away from what looks familiar. So these sloths here on the other side, that's what, if you lived 10,000 years ago, as obviously some humans did, that's what you would think of if somebody asked you to draw a picture of a sloth, right? That was your average sloth back then. What we have today are very unusual sloths, actually. And so we know that the vast majority of life is extinct, and we know that the vast majority of variation is extinct. And so we want to be able to understand how that variation arose and what processes gave rise to that variation as well. And so this is what we've been doing for many years in my lab. And actually what we tend to do is try to build these really big 3D image databases using things like laser scanning, like in this image here of a dinosaur at somewhere in Canada, I think. Yeah, I think it is the Royal Tyrrell in Canada. Or we use CT scanning to try and build up a really nice comparative database of 3D anatomy mainly for vertebrates, although I dabble in other clades sometimes. And so now we have thousands of species in our data set. We can start to try and reconstruct how this diversity of life evolved. Now we run into a lot of issues right away with this, which is that we have a lot of variety of different forms, and kind of standard methods don't really work when you start to try and compare, like a frog to a whale to, a T-Rex. And what we want, because we have these really, with all the new technology that's available to us, we can get these incredible images. And so we don't want to just dump that out for kind of standard methods that people used to use. If we did do that, it would give us something like this. So these are what we call type 1, type 2 homologous landmarks. These are points of correspondence across the different skulls. And if we were to limit them to things that we absolutely were sure were the exact same point, which actually, to be fair, we're never sure, but we like to believe so. So for example, where the premaxilla and maxilla meet, or things like that. If we looked across all of our species in our data set, we can only find about 12 points, which really does not give you can see where these points are in the skull, you're leaving out so much of the variation that it's really unsatisfying, and you're going to lose most of the diversity if you do it that way. You can tell some things. So this is if we take those 12 points for our thousands of species, this is a very dirty plot because not great data. This is basically what we can tell. So this is a morphospace again. And what this does is take all of our data and it summarizes it into just a few kind of major axes of variation. And each axis here represents a combination of our original traits. And so what we can see across our entire data set is that birds are weird. I mean, it took us several years and a lot of money to come to that conclusion. But to be fair, it really overturned something that I've always believed, which is that birds are boring. Sorry, I'm kidding. Birds are not boring, but we'll come back to that later. But birds are very unusual, and all the other groups of vertebrates, or tetrapods anyway, are fairly similar in many ways. And then, we can do things like try to morph a skull between these different axes. So this is where I take a fox skull. Fox is the most average mammal according to our analyses. If you morph it to a bird, you can get something horrific, like on the side here. On the other end, it looks more like a normal animal, but you can't really do that much more with this kind of data. And so one of the things that we really spend a lot of time on is how can we get more from all the incredible data that we have available to us for all these species. I work at the Natural History Museum. We have, some 90 million, give or take, specimens. We want to do better than put 12 dots on them, right? We want to actually get some realistic description of their shape. And so we came up with this approach, which is a semi-landmarking approach, where we can put all these dots onto this really simple template and then actually map them onto all of our specimens. And when we do that, and this is really, really difficult, really time-consuming, extremely tedious, but when we do it, we end up with something I think is pretty glorious, which is this really nice description. So in this case, all these points here are 3D descriptors of the shape of the skull. And then we can analyze those to look at how that shape has evolved. And so we can take that then and try to answer this, dispassionate question of why do animals look the way that they do? And so we've done this for many, many species over the years. We've looked at things like squamates, and we've used our understanding of how they're related to each other, this phylogenetic tree, and try to estimate how quickly they've evolved on different branches of that tree. So in this case, the different colors in this little plot here represent faster or slower rates of evolution. We can look at how different habitats influence how things have evolved. So this is a study we did of crocodiles, where we showed how aquatic to terrestrialization of different clades of crocodiles is actually the primary driver of their skull shape. We can look at things like birds and their relatives, their closest relatives, the non-avian dinosaurs. And we can access things like how different developmental tissues that form the different parts of the skull actually have a strong impact on how the different parts of the skull have evolved. And we can also look at comparisons across the different clades and how quickly they evolve, which I'll show you in a bit more detail. We can look at things like salamanders and frogs, some of the different metamorphic groups that have evolved, and look at how those life history shifts have influenced their evolution. And we can, of course, look at our own group. We can look at mammals, and we can look at not just their development and their ecology, but we can even look at things like social structure and activity to understand how those have impacted how they evolved over the years. And so through this approach of biodiversity phenomics, we can really access an incredible diversity of topics. And this is just a snapshot of some of the things that we have done or do in my lab using this approach. Now, of course, I would love to be able to talk at length about every single one, but I don't have time. And so I'll refer back to the closing line of that initial description of evolution, which I think is another really great way of putting things. I'm going to offer you today a very imperfect and incomplete sketch, as much as I can do in such short a space, but I'm sure your imaginations can fill up the wide blanks, or you can also ask me over wine later. But I'm going to just give you kind of 3 different examples of some of the work that we've done and how it kind of shows some of the interesting ways that we can try to understand the factors that have really shaped the evolution of these different groups. So I'll start off with the birds and dinosaurs. So as I mentioned, the skull is made up of a couple of different tissues, the neural crest and paraxial mesoderm. And it's often been suggested that the neural crest is the key innovation for vertebrates, that it is just far more capable of evolving unusual shapes and unusual forms than the mesoderm was. And that's why we have such incredible variety within vertebrates. And actually, so we can test this hypothesis with our data. And so what we see here is an analysis that we did of the bird skull. And the yellow parts of the skull here are the anterior cranial neural crest. The dark green are the posterior cranial neural crest streams, and the light green are mesoderm, which again is kind of that ancestral tissue that everybody seems to feel like is a bit rubbish, to put it scientifically. And what we see, in fact, is that we do see a faster rate of evolution in the parts of the skull that are formed by anterior cranial neural crest, in particular ones that are formed by that anterior stream, but also ones that are formed by amalgamations of different bones. So complex development seems to lead to complex evolution. And so that was quite an interesting, I think, first real macroevolutionary test of this developmental hypothesis, that development leaves a mark. Now we can also take this amazing bird data set we had 350 some odd species. We can add in dinosaurs, again, because we have this nice new way of capturing the shape of the skull. So this is another one of these morphospace plots. What you can see in the silhouettes in the back are the kind of shapes that are represented in the different parts of this morphospace. The black dots are birds, and hopefully you can see, but there's colored dots in there that represent the non-avian dinosaurs. And while there are far more birds in our data set, because obviously preservation makes it harder to get nice 3D skulls of dinosaurs, the dinosaurs that we do have actually cover a huge amount of the area of morphospace relative to birds. So we can then take this data and plot it onto a tree of the relationships among these different groups. And we can look at the different parts of the skull in turn, or we can look at the whole thing together. And what we actually find is that, again, this is one of these plots where we have the tree and the different colored branches represent how fast things are evolving. So red is fast, blue is cold, and or blue is slow. And we can see that in a lot of these clades, the fastest branches are actually in the dinosaurs. And this is after we correct for all the different ways that we could be affecting this kind of data with things like how long the tree branches are in our sampling and things like that. And in fact, when we take all the different parts of the skull, what we find is that birds evolve much more slowly than all of the non-avian dinosaurs. So despite what I said earlier about birds being extremely weird, they are weird. And on that branch leading up to birds, which we don't have a very good sample for because birds don't preserve very nicely in the fossil record, there must have been a really dramatic shift. But once you get into birds, once you are a bird, you actually really hit the brake pedals on how quickly you evolve. And birds really evolve far, far more slowly than what we see across other dinosaurs. Now, that may feel counterintuitive, but actually it makes a lot of sense. Because when you think about what birds do with their skulls compared to what other dinosaurs do with their skulls, there's not nearly as much happening. When you think about the variety of dinosaurs, we have dinosaurs that have incredibly complex chewing. We have dinosaurs with huge ornamentation on their skulls, like Triceratops. We have dinosaurs with these big bossa on their skulls for fighting. We have dinosaurs that do display, that have lots of different kinds of prey capture behavior, all kinds of different things going on in the skulls of dinosaurs. What do we have in bird skulls? We have a really solid brain case structure to protect their big brains, and we have a beak. Birds don't do complex chewing. They've really reduced all of those kinds of jaw musculature aspects that are really elaborated in other dinosaurs. And all the really kind of display things that we see, with some exceptions, of course, but overwhelmingly, they do that with feathers. So the things that dinosaurs did with their bony elements, birds actually do with soft tissue. So it really makes sense that when we're looking at just the skull, birds actually really slow down. And actually, there was a paper, I think, just in the last week that shows the same thing when you look at the postcrania of birds, too. When you look at their limb skeleton, once they get into being birds, they really slow down evolutionarily. But that's something that's really hard to tell if you don't have a way of putting all this into the same kind of data set. Now, so I'm going to shift tack a little bit and talk about salamanders, which is a group that, as a mammologist, I tend to look down on all of the other clades. But upon reflection, I can admit that salamanders and amphibians in general are the most interesting of vertebrates. And salamanders, and particularly so, because they show this incredible diversity of developmental mode. And so what we see when we look at salamanders in the same way, again, capturing their skulls in really nice detail, is that the major thing that distinguishes different kinds of salamanders in terms of their skull morphology, or their shape of their skulls, is how they develop. So whether they're direct developers, whether they go through metamorphosis, or whether they're paedomorphic and stay in that juvenile form, they have incredible differences in their skull morphology based on just those things. We can again do the same thing where we map how fast their skulls evolve. And what we find is every time they shift in their life history strategy, which happens many times across salamanders, they go from metamorphic to paedomorphic, they go from back and forth from direct developers to metamorphic forms. Whenever we see those shifts, we see a change in the rate of evolution. So life history is incredibly important for salamanders. And in fact, we find that the direct developers are really slow evolving compared to the paedomorphic and metamorphic forms. So that gives us some new information that we can then put into models for how species might respond to, for example, climate change or environmental disturbance, because we have some understanding of what factors affect how they evolve or how quickly they can evolve or adapt. Now, we had thought we'd see the same thing for frogs, because obviously frogs are all metamorphic. So we thought, the ones that have kind of the most extreme metamorphosis or the most extreme differences between life history stages would also show the fastest rates of evolution. And this is something that's actually been really widely debated, I think, in terms of the evolution of metamorphosis, is what impact does it have? And what are the benefits and costs of metamorphosis? And what we found, actually, surprisingly, is when we look at frogs that have really active larval forms, so completely independent tadpoles that are living a completely different lifestyle than the adult forms. And we compare that to what we call a direct developer, which in a frog doesn't mean quite the same thing, but means that their larvae are non-feeding, so they're not going out and getting their own food and living independently. We find that the direct developers, the ones who kind of lost this kind of separate active larval phase, evolve much more quickly. So even though you have this incredibly rapid or extreme metamorphosis, this disconnect in life history stages, it clearly still has a mark. It leaves an impact on the evolution of the adult skull morphology. And that is something I think that is quite surprising. Okay, and then finally, we're going to get to what I would long have believed is the best group of vertebrates, which are the mammals. Mammals are an amazing group in terms of their diversity. We obviously know quite a lot about them, and it gives us a lot of opportunity to do really interesting things with mammal evolution. And so last year we published this paper, it's got the NHM on the cover, which is really nice, about the evolution of mammals through the Cenozoic. And for that, we again gathered this really nice data set capturing skull shape in living and fossils we are able to get a much nicer data set of fossils for the mammals than we could for any other group, because mammals have an amazing fossil record. But interestingly, what we found is that mammals have an incredible amount of convergence in their skulls. So this plot here, it's again another kind of morphospace, another principal components analysis, to use a precise term. But in this case, the colors here actually represent the density of different species occupying the same area of morphospace. So the yellow area, that kind of really bright spot in the middle, is an area where you have tons of different species evolving the same morphology. And if you looked at the different kinds of mammals that are out there, things like the Afrotherian mammals like elephants and elephant shrews and sirenians, if you looked for xenarthrans, things like sloths and anteaters and armadillos, and if you look for the kind of the northern radiation, so primates or cats and dogs and ungulates, you'd find representatives of all of those groups in that kind of hot center of placental mammal morphospace. And really the only ones that stick out are the whales, as you can see, and the rodents also stick out as somewhat unusual in terms of what they do with their skull bones. But really mammals are incredible in their amount of convergence. Now we can take all of that also, and we can start to look at what kind of factors are really influencing how mammals evolve. What are the dominant factors influencing how our clade has evolved the diversity that it has? And again, we do this using our understanding of relationships and their different traits, and we can do far more with this because we actually know so much more about the ecology of our group. And some of the things I thought were particularly interesting that came out of this research was that development has a huge impact on how mammals evolve. So precocial species that have relatively little parental care when they're born, that when they're born, they can immediately get up and run away from a cheetah or whatever, the ones that are in every nature documentary getting killed. They evolve really fast compared to ones that have really prolonged parental care, like our group. Social mammals evolve far faster than solitary mammals. And the thing I always point out when I mention this to people is that I did this entire analysis during the pandemic. And so that, felt a really harsh thing to point out to that point, because I think we all felt that kind of reduction in evolution during that period. And also things that don't have a fixed activity pattern. Maybe this is also relevant for the pandemic. You're not necessarily awake only during the day or only during the night, but you can kind of modify based on your situation. They also tend to evolve faster than other kinds of mammals. And so these features are things that we also want to then start plugging into the predictive modeling about how different species will respond to kind of changes in their environment. Now, again, we can take all of this, and this is all really based on our understanding of the relationships of these different taxa. And we can plot them on a tree like this one. This is our mammal data set. And again, the red branches are the ones that are evolving really quickly. And so I have the whales pointed out there because they show the fastest evolution. But some of the other ones do too. Actually, the early parts of those trees show quite a lot of evolution to the base of a lot of the major clades of mammals. Now, this is, of course, a time-calibrated tree, which means that we can take this and start to ask questions about how species have changed, how the rate of evolution has changed through time, and not just along the tree of life. And one of the questions I've always been really interested in with mammals, and this is also driven my field work that I mentioned at the beginning, is really how they responded to end Cretaceous mass extinction. Now, there's a lot of nuance in this discussion and a lot of uncertainty, but we can basically take these estimations of rates of evolution and put them against time. And so this is where we enter a very experimental phase of this talk, really, which is we were trying to figure out novel ways of communicating these sort of patterns to broader audiences. And one of my postdocs, Aki Watanabe, had the brilliant idea of putting them to music. And so you get to experience what evolution sounds like by listening to first the rates of evolution for placental mammals through time. There is a slight lag, I think, because of getting over Zoom. You can see you're moving forward through time. That high point there was actually the evolution of whales. So there was a really rapid burst of evolution of whales. But what we can see actually with placental mammals is that the rate of evolution has really declined through time. It's been slowing down ever since that initial burst of radiations as a KPG mass extinction. But it hasn't just slowed down in a kind of uniform way, what people would often think of as what we call an early burst model, where you just a burst of activity and then a decline through time. We see a lot of later bursts. And so what we coined here was a new model called the attenuated model, being a weakening wave through time, which I think more accurately describes how most groups of organisms probably evolve. You have this initial burst of diversification and then a decline, but you have later bursts that probably reflect major environmental changes. So maybe they represent rapid periods of global warming, which we know we've experienced in the Cenozoic, not nearly as fast as today, but relatively fast. Big changes in cooling, the onset of seasonality. We can pick out some of these patterns in these bursts that we see later on in the Cenozoic. Now, do we see this in some of the other groups? So here we are with birds. And again, starting off with what I think are, this really shift from early birds that were more like dinosaurs. And what we see with birds also actually is actually decline through time. We have this initial radiation. We get bursts later with some of the big radiation that's really taking off, like the passeriforms, but you see a slowdown through time. What about some of the classic groups that don't do anything? Let's look at crocodiles. Are they in fact the slow and steady group that we think of them being? We talk about them as living fossils, right? That they never really change through time. Kind of feels like that right now, right? But actually, I think crocodiles are underappreciated, or the group during the Cretaceous in particular. They really evolved an incredible variety of terrestrial forms. They were relatively small, they were herbivorous, and they were adorable. And I think they would have been absolutely fantastic pets. So these are the ones that unfortunately went extinct, as did a lot of other things in that end Cretaceous mass extinction marked by the meteor there. But actually, what was surprising is that in our analyses, crocodiles are actually really increasing in rates of evolution to this day, especially in the kind of Australasian region. You see lots of actually rapid rates of evolution. They're re-evolving the same things over and over, but they're actually evolving quite fast within that area. And so it's still, I wouldn't count crocodiles out. And I was just in Queensland, so I'd also not go swimming. Of course, we're talking about the ones who are still around to talk about this, but there are groups that didn't fare so well going through mass extinction. So you can also look at this, use this kind of approach to think about, whether there was a period of decline or not in advance of a mass extinction. And this is an area that a lot of people have really debated. So, with dinosaurs, we can see some of the signatures of the rapid burst. For example, we see lots of diversification of sauropods. We see, the diversification of some of the groups that people really focus on, things like triceratops and some of the things that people enjoy. But we see this increase in rate of evolution actually right before the mass extinction. And so it cuts off really suddenly there, of course, because a meteor hit the earth and dinosaurs go extinct, not even dinosaurs, of course. But at least from our data set, it doesn't look like things were slowing down or things were getting rubbish for various reasons. It actually seems like they were still charging ahead at full speed when they were cut short. And so we can put all of this together, and this is definitely the most experimental part here, where we try to put all of our different data sets together and see if we can see any commonalities, but also just, I think, interestingly, try to hear the symphony of different species coming in. So hopefully this will start imminently. As I said, this is quite experimental, so there's maybe a couple of things in there that slightly out of place, but I think hearing all of them together is an interesting experience. So again, here we have the crocodiles, dinosaurs, turtles, lizards, frogs, salamanders. We're going through the Jurassic into the Cretaceous. See some of the other groups starting to appear, so we get the snakes really coming in and do some things that we have around today, figuring out to a certain extent. Then we start to get into some of our beloveds. We lose the dinosaurs, of course, but we get the mammals, big numbers, and then we can start to see that pattern there. So it cuts off quite suddenly, of course, because we get to the modern day. And so on the one hand, it's not the same as what was going on with our dinosaur cutoff because we've just gotten to it today, but hopefully you can all hear that there's a lot happening. It's, it's still very upbeat. There's some clades that are showing some really nice increases in diversity. There's a lot of, as I mentioned, even with things like the crocodiles, there is a lot of diversification happening in these groups. So very different in one sense from what was going on with the dinosaurs, where we just cut them off out of nowhere. On the other hand, we all know that that's exactly what's happening, right? So here we are at the modern day, we have all these groups that are still diversifying in lots of interesting ways, and yet we see these now very dramatic declines in Earth's biodiversity. And so, of course, we know what's causing this. We know that it comes down to human caused factors, right? These, you can see from the studies, from all the comparisons to those trends within their clades, that you don't get a rapid cutoff without something dramatic happening, right? And so we are obviously in the middle of something incredibly dramatic with obviously climate change, global warming, human caused habitat destruction, pollution, poaching, everything else, really driving the decline of Earth's biodiversity. And so, when we think about extinction in the past and the incredible impact it has had on the diversity of life on the planet, and we can look at where we are today and these really, really sad indicators of what we're doing to life on this planet, we have to reflect on the fact that, of course, we are the meteor today, and we are the ones that are causing all of this. And here at the society, for 235 million years, we have gathered to talk about our passion for the natural world and share our experience, our knowledge, and our understanding of the natural world. It's not enough anymore. It's been okay for a couple of centuries, but actually, I think we can all appreciate that it's not enough anymore. And I hope going forward, we can all agree that we need to not just talk about the natural world, but really we need to start acting far more strongly to protect the natural world, lest we think of all these things as just species that occurred in the past, right? And so if we don't, I think, really start to increase the activity that we do as individuals, but also as a society, right? We have incredible global reach. We have thousands of members, thousands of fellows across this planet, including all of you in this room, all of you joining us online. We have incredible capacity to make change and not just meet here to talk about our love of the natural world, but actually to act to preserve it. Because if we don't do that, if we don't really seize this moment to really make dramatic change and force dramatic change, we're not going to have anything left to talk about when we meet here together. And so I will leave you with this final thought by Darwin again, which I think hopefully we will all take as a call to arms to really see the society harness its potential, harness our incredible assets, and all of us as fellows of the society to do what we can, not just to come and meet with each other, but use our incredible networks and our abilities to really make change for nature on the planet today. Thank you.
So thank you for that amazing, stimulating, thoughtful, interesting, provoking talk, Anjali. It was really brilliant. So what we have now is 10 minutes for questions. Okay.
I'll take a peek if there's any online as well. Yeah. Will do. Hi.
So, thank you for so then let's imagine that we manage to stabilize climate change, so diversity. Can we predict like a new burst in diversification or acceleration of the rate of evolution for the next million years?
I mean, I think we probably can. I think even if we do fail to stop the decline of biodiversity, there are clades that will benefit, right? There are environments that will expand and certain groups will probably increase numbers and diversify, it'll just be a very, very different world, right? And so, yeah, I think absolutely we will see something probably proportionate. It'll be proportionate to kind of the emptying of ecospace, right? So you see these big bursts after mass extinctions because there's so little morphospace, so little ecospace, or so much ecospace available. And the reason those later bursts are smaller in amplitude is because there isn't as much new ecospace available. And a lot of times they'll be associated with changes in climate that create new environments. Sometimes they're associated with moves into completely novel environments, like when whales move into the water or bats move into the air, just talking about mammals specifically. So I think we will, probably, if everything stabilizes, see some diversification, filling in some of the ecospaces that have been left empty by extinctions to this point. But I think the lesson the thing I often tell people when we talk about extinctions, because as a paleontologist that talks about mass extinction a lot, we look for patterns, right? We look for patterns, what's going to do what better, what's going to do worse. We try and find commonalities, like big things do worse than small things with low population numbers do worse than things with big population numbers. This kind of ecology will do better than this kind of ecology. Those things hold true to a certain extent. Those are really based on kind of background extinction, which, is just the stuff that's happening all the time. When you get into mass extinction territory, all bets are off. It starts to become a lottery. We tend to focus on the winners, the things that make it through the last mass extinction. But what we forget is that most species of mammals went extinct. Most species of birds went extinct. It was a squeaker for all of us. So I vote for not risking it.
I just wondered if you had any idea of any of the spatial or the diversity of cells and kind of, the fucking returning with the other species.
Yeah. Oh, I'm sorry, I didn't repeat. You're right. So asking about why crocodiles really seem to start to show this diversification more recently, whereas some of the other clades maybe are more kind of in decline. So I think it's not entirely clear, but I think it may well be that, in this specific environment of Australasia, because it's really those species that where you see kind of more recent kind of diversification and change. What's interesting is that they're not actually really inventing novel morphologies. They're just kind of re-evolving similar things over and over. So it probably has to do with, biogeographic patterns in that specific area more than anything else. Whereas the other clades, their patterns are far more kind of global.
You mentioned that the evolution of the skull of birds over time has not changed very much and it's been rather conservative. But I would think that if you include the bills of birds, you will see a very different picture. What are your thoughts about that?
Everybody else. So the question is about the skull evolution in birds and whether bills would make a difference. So we do include bills in that analysis. Well, we include the bony skeleton underneath them, which captures at least the major changes in terms of length.
Sorry, the bills are not part of the bony skull.
No, but the part underneath the bill, the rostrum, is capturing, and it'll capture the vast majority of the morphology of the bill. There will be some kind of keratinous aspects that aren't captured, but largely you're going to capture the major variation in the bill, which is, overwhelmingly kind of a length focused thing. There's, major differences in length. There's some differences in width, and that's true. That part of the skull is by far the fastest evolving part of the skull in birds. It's still not faster than that same part of the skull evolves in dinosaurs. Because if you look at non-avian dinosaurs and what they do with their rostrum, right? They don't just kind of move along this one major axis of variation and a little bit on another one. They're doing all kinds of crazy things with their front of their skulls. Again, they're having really complex mastication, all kinds of crazy jaw musculature is evolving. They do lots of really interesting ornamentation on the front of their skull, on that rostrum, that same part of this bony skeleton that is underlying the bill in birds. And they even do things, there's species of dinosaurs that I can't tell the front of the skull from the back of the skull because they're so heavily modified. city potty, for those who know, I can never find the front. But so I think although birds are incredibly interesting and diverse, they're not, and that pains me to say because I actually really don't like dinosaurs, but they're not as interesting or diverse as non-apian dinosaurs, certainly in terms of skull morphology, in the rostrum as well as all the other parts of the skull. It's actually on one of the plots. I have the rostrum picked out in specific, but I won't bother. Maybe I'll show you another time.
Thanks for that. Lecture. Very early on, you showed a slide that is measuring number of families that are diversity. Kind, but if families are arbitrary units, they're not absolutely comparable. How does that work?
Sacrilege in this house. So the question was, given that families are kind of unequally named in different clades, how can you compare families through time across? So I think you're talking about the very, very beginning. I think that was marine invertebrate families through time. This is a classic study by Jack Sapkowski, where he put together this massive compendium of marine invertebrates. And so, yes, I think you would struggle. I would not compare diversity through time in vertebrate clades. Obviously, if you look at the number of orders of amphibians versus the number of orders of placental mammals, there's no comparison between those things. Genera, on the other hand, tend to be a little bit more reasonably or consistently applied across clades. So that's, I think, the more reasonable one. But that's not my plot. That's a plot from, quite frankly, the 70s. But we need to start somewhere, right? And so I think species are difficult to do in the fossil record. There was a good study by Dave Jablonski, who was last year's Darwin Wallace Medal winner. Don't forget to nominate my favorite scientist. And he had a paper about a decade ago in PNAS looking at the validity of morphogenera for macroevolutionary studies and found that those were quite stable. But you're absolutely right. I would not compare numbers of families across widely dissimilar clades because they're not equally applied. I think there was questions that were on that side. I think John's had his hand up for a while. You didn't complain about birds.
What you did in the orders and let's declassify this. Then you try to do a similar type of analysis in parts. Firstly, how much of that degradation benefit down to different ways it comes to.
Yeah, so that's a good question. There are people who are looking at this. I think there's a group up in Edinburgh in particular, and a group in Bristol who are looking at this in terms of developmental patterning and how it dictates why there's, I think one of the questions looking at things like Fibonacci sequence spirals and why they're so ubiquitous and why you get this really constrained kind of patterning within plants. I don't think that there's anything similar within plants because, and I've occasionally thought about plants and then decided to do something easier like insects. Because what part of plants are you going to look at, right? Plants are so incredibly modular. Are you going to look at flowers, leaves, the way they're preserved? One of the biggest issues is getting things in the same format. Now, people have looked at like leaf shape, for example, and that's been commonly applied mainly for things like environmental estimation. Oh, sorry, the question was, has anybody done this in plants? I didn't say it already. But not necessarily so much on the macroevolutionary scale like this. I think it's a really interesting thing to try and do. And my guess is you'll see something similar in terms of basic developmental patterning. I think people have approached it in a discrete way in terms of looking at different ways of growing and kind of discrete characters that you can look at plant morphospace in that way, but not really from a morphometric perspective at the same scale. But I think it's a really interesting thing to do. It's just a very daunting challenge with plants.
And how does that contribute to how we have people, you know, change the kind of
So the question is about the genetic diversity of cheetahs and whether their kind of famously low genetic diversity will affect their ability to kind of escape extinction or survive. I'll confess this is quite far away from my area of expertise, but it's certainly something that people talk about quite a lot. One thing that I've heard recently that I think is an interesting thing to consider is that we don't necessarily know what is a normal range of variation for genetic diversity across taxa. People focus on a couple of key species, things like cheetahs, but there are probably many, in fact, there are definitely many clades around that actually naturally have low genetic diversity and always have, and have actually maintained that for millennia, if not millions of years. And so I think before we decide how genetic diversity is going to impact survivorship, I think we have to consider everything, because obviously we're running out of time to answer these questions, and so we can't necessarily dive into the level of detail that we would want to for each of them. But I think we don't actually necessarily have a good baseline for what is genetic diversity supposed to look like, and what is unusual and what is just normal for a certain kind of organism.
On plants, there was actually a very good study a couple of weeks ago in Nature Plants by Convergences group. I'm sure it's not Joe.
I did see that one though, yes.
Just back up. It seems that the really interesting question in evolution is about the origin of the major groups themselves. Has your research on skull shed any light on the origin of the major groups?
On the origin of the major groups themselves. So the question is whether our studies of skull evolution have shed any light on the origin of the major groups themselves. So I think, with the placental mammal study in particular, that's something that we've been trying to focus on, is whether we can see any major shift in the evolution of skull shape. Where I think the most kind of generalizable result in terms of skull evolution from our work is really looking at that neural crest issue, because it is something that has been kind of thrown out there as a hypothesis, and has sort of become just commonly appreciated or accepted that neural crest is the source of all diversity, and the reason that you have such incredible diversity in vertebrates is because of the evolution of neural crest. And what we have seen in all of our studies actually is that does seem to be the case. One thing I didn't talk about here is that we can map which parts of the skull are evolving the fastest, but also which ones are the most variable. And when we look across all the vertebrate groups, it's not always in the same place. Birds and mammals tend to have the most variation in their rostrums, in the anterior part of their skull. Squamates and amphibians tend to show the greatest variation around their jaw joints, but both of those are neural crest derived regions. And so what we need to do now, and what we plan on doing now, is actually really trying to dive into the development of those organisms and figuring out when in development do you see those major divergences happening. Because I think now that we know where the variation is actually concentrated, now we can get at the actual mechanisms that gave rise to them. So I think that's probably the most relevant in terms of the origin of the diversity of the major clades themselves and why they follow specific paths in terms of this. But it's still a wide open question. We have the pattern. What we don't know is the process. That's usually where it goes for macroevolution research, right? Because until we have that pattern, we don't know where to look. But now that we've actually pinpointed a couple of key regions that discriminate the major groups of vertebrates in terms of how their skulls vary, we can look at that in more detail. I mean, what we have also done, of course, with all of our data sets is reconstruct an estimated ancestor for all the major groups and what that ancestor looked like. And then we've inferred the ecology of that different group. So, what would the earliest placental mammal have looked like? What would the earliest bird have looked like? And that gives us a search image for fossils, but also gives us some indication of what the ecology is of that most recent common ancestor for those groups is. So I think that's way more in line with what you're thinking in terms of the origin of those groups. But you're right about the Donahue paper. It's in my to read folder. I'll get around to it.
So at this point, very sadly, we have to say goodbye to all the people that have been joining us online, and thank you very much for being able to do so.